Okay, thank you very much. I hope you can all see my slides. Thanks for inviting me to give a presentation at this wonderful event. Um, I'd like to say that, the reason why we started this work on the conserved domain database was partially, going back to our experiences with the CASP experiments. Because one of the things we found out when we analyzed why, we did not make correct predictions in cases that were rather easy and straightforward is that we had a template library that was overly constrained. So we concluded, well, we don't understand these protein families well enough. So, we need a better way to model them. And, with the development of our PS blast, which is sort of the reverse position specific blast that happened at the same time, it seemed actually quite straightforward to start compiling a template library, which essentially are protein family models. And, let us characterize and describe what's conserved in protein families and, hopefully identify more and more diverse members of these families in database searches. So protein family models for us are essentially multiple sequence alignments that we can turn into statistical models, such as PSSMs or HMMs, and make them subject to search via, query protein sequences. And they have a variety of uses, right? And that support all kinds of work that biologists are interested in doing. Now, about 3D structure, so biologists are more interested in linear annotation. That means, they want to know where are the domain footprints on the gene product and, where are functional sites if we can identify them. And these were the things we sort of set out to provide. But our goal was to have, 3D structure being a main driver of the family models that we generate. So the planning for CDD actually started about 25 years ago in mid-1999. I think we had the first public release of sort of a small, minuscule search set sometime a year later in the summer of 2000. The first paper came out 2002, and we were going since then. So what we sort of planned to do back then was to actually do manual curation on protein family models. That seemed necessary because the functional information we wanted to capture was relatively complex and often just found in the published literature, and we had no automated ways to obtain that. And we also wanted to record the site of, conserved function in those protein family models so that things like cofactor binding sites, active site residues could be mapped onto protein sequences and people could use them in their research or, forming hypotheses of, why sequence variation, for example, was, deleterious in an organism. And we thought, we'll get a lot of this from actually using 3D structure data. So we had, at that time, the molecular modeling database, which was NCBI's and still is mirror of the PDB collection. We had a very rich data set of pre-computed 3D structure alignments, computed with the VAST algorithm that was developed by Tom Mayday, Jean-François Gibrat, and Steve Bryan at that time. And VAST is still active and still being used by us. And we had the beginnings of a 3D structure viewer called ISMB that could be used to visualize 3D structure and structure superposition. So one thing we noticed from the beginning was that the accuracy of the existing seed models that we found in databases like PFAM was really great. But when we looked at structure pairs in these alignments that are more diverse, that have relatively low percentage identity in structure alignments, the agreement between the multiple sequence alignments that we downloaded for initial analysis and the vast structure superposition was not very good. So we thought maybe we can do better and improve those models, particularly when they try to describe very diverse families. And we could show at that time that correcting imported alignments would actually result in statistical models that performed a little better. They were more sensitive and they produced more accurate alignments down the line. We also at that time looked at the COG resource, the cluster of orthologous genes that was developed by Eugene Koonin's group, and resolved protein family membership, at a very granular, fine-grained level. And it turned out that biologists were really interested in that. So we set out to build CDD into a resource that doesn't just, model domain superfamilies, but actually tries to model functional hierarchies of the diverse superfamilies where we could eventually assign very precise function to individual subfamilies and make that resource available to the biologists. So since we started to go down the route of having curators work on the resource, we sort of understood at some point that, we need to build an infrastructure that is fun to use. And, fun in that sense means it's easy to use. So the learning curve is not super steep. Ideally, we'd have curation tools that are applications that run on personal computers. They don't require, knowledge about installing and maintaining software by the curators. That, reduce sort of repetitive stress and fatigue on the curation team. And, that are powerful enough at the same time to sort of enable biological discovery, right? So we ended up with building the ISMB molecular structure viewer into a very powerful multiple sequence alignment editor, where you could see, multiple structures superimposed, plus the corresponding sequence alignments, plus many, many more sequences from GenBank and other sources, added to those models, up to, several thousands of sequences that, the alignment editor could handle easily. And then we had sort of an overarching tool called CD3 that allowed us to track the models, interact with the model tracking system, and all of these kind of hidden from the curators, right? The curators just we want to check out a model and they want to work on it and say, well, I'm done with it, and, and move on to the next thing without having to worry much about the technical details behind the whole system. One thing we also found out was fairly crucial is that we had sort of an open discussion set up between curators and also to some extent users of the database, where models and the strategies to build those models and those hierarchical classifications were criticized by others, and where curators sort of mutually correct their work or validate it. While this was a little bit contentious in the beginning, it turned out that it improved the accuracy of the systems that we built significantly. And, whenever we tried to reduce the number of validation by others, that we required in the system, we saw accuracy drop down a little bit. I mean, it's just human nature, but, that was an interesting thing to learn. So this system supports public services, and has been running for quite a while. And more recently, we were sort of drafted into expanding the curation work that we do into curating protein domain architecture. So an architecture is sort of a succession of several conserved domains on a protein sequence. It can be one, it can be many. And what the curation work we're doing in this architecture space is we suggest names for domain architectures. And in cases where we have collections of sequences that do not have submitter provided names, in the RefSeq database, we're using these domain architectures currently to name about 35% of the bacterial RefSeq proteins. The majority of the proteins are named by things other than domain architecture. So there is, high similarity to UniProt sequences, for example, or HMMs from PFEM and other sources that are used in the process. But sort of at the lower level where we say, well, a more generic name is better than nothing, and just saying, well, this is a hypothetical protein, we don't know what it is. If we can say, well, it's a ubiquitin family protein, for example, that's some information, right? That's better than having nothing. So this work recently was also extended to add transferable attributes to the architectures and map them onto sequences such as Go terms, EC numbers, etc. And we've now started a project in the past few years to expand the coverage to small eukaryote assembly proteins. So this is fungi, this is protists, many human pathogens in that set, and also nematodes. And this is work that supports NIH's comparative genomic resource and is aimed at, providing tools at the end of the day that let submitters sort of put a minimum standard annotation on genomes in those taxonomic realms, basically. So we tracked for the longest time, what is the coverage of CDD of proteins with known 3D structure, and sort of we managed to stay sort of in the range of 95 to 98%, and families with known structure were very attractive curation targets because the structure informs us about what should the shape of the alignment be, right? What are the things we expect to be conserved and present in all family members, and where is length variation in this family? And then, AlphaFold happened, and we have a lot more structures available than we ever had before. So the question was now, well, how can we use all this, rich information? And, one of the obvious things to do was to sort of point users of BLAST to the case that, we can map many of their query sequence through the sequences they hit in the BLAST database to AlphaFold structures. And iCn3D came in really handy to sort of directly allow visualization of alignments between user queries and predicted 3D structure, and sometimes also experimentally determined 3D structure directly out of the BLAST results display. So what CDD curators are doing is, they will look at AlphaFold predicted structures if there is no experimental structure available in a domain family or protein family. And iCn3D is extremely convenient for that because not only can you visualize what the structure is and see how, the position of helices, strands, conserved, core elements you would want to preserve, how this relates to your alignment model, but you could also launch similarity searches and then look at, eventually multiple structures, that could be similar to the family that you're interested in. And by that, you could also get information about function. So we sort of turned the workflow around, and we now actually tend to work by creating lots of candidate models automatically, and have large data processing in place where we could extract information about models with respect to function from various community provided resources, but also from taking AlphaFold predictions and doing vast searches of those against the database of experimentally determined structures, and presenting this again in a nice triage interface to our curators where they can see whether, a novel domain family actually has, 3D structure neighbors that might be meaningful when we search with the AlphaFold predicted model.
Hi, Aaron. Can you wrap up, please?
Yes. So currently, we're involved in characterizing additional protein domain families. We want to extend coverage, of course, but more importantly, improve resolution and the accuracy. And we hope to be able to use, all of this richness that we get from predicted 3D structure for this purpose. And ideally, we'd want support for modeling protein families built into iCn3D. So this is sort of the next goal. And we're also asking ourselves, because we're dealing with sustainability of our resources, whether we actually need to pre-compute 3D structure comparison results in a vast database, which is great to have. It's a treasure trove of information. There's, literally billions of alignments, but it's very costly to maintain this. So if you're interested in providing feedback about VAST, if you're using it and it's useful to you, please contact us, because we're organizing some interview sessions in the next few weeks, and it'd be nice to have more participants. Thank you, everybody.
Thanks, Aaron. So, are there any questions from the audience? We have time for one question at least.
So you mentioned AlphaFold, and I'm wondering if you have ideas or plans to try to discover new domains out there in that deluge of data that we have displayed.
Yeah, we're sort of glancing at the efforts to cluster the AlphaFold predictions, that were done through FoldSeek, I think, and how we could utilize those sets of clusters to filter out promising candidates, right, for novel domain families that we haven't seen so far. It's getting really difficult actually to increase coverage. So both PFAM and places like CDD actually work really hard to keep a coverage rate of sort of 86-85% or so of known proteins, right, with family models that we have. And we're sort of exploring the tail of the distribution, right, where many of the new families that we define are kind of taxonomically limited and have very, very narrow coverage. What we also see when we use 3D structure, though, is many of the novel domain families are actually similar to, large superfamilies that we know about. We just didn't see that similarity when we did sequence-based searches.
All right, thanks, Aaron. Yeah.